Hello, everyone. Welcome to section 2 of the Design Computation Input Output 21. This will be about computational design and cognitions. And it is a real pleasure to have Professor Jeffery here today. Professor Jeffery is interested in how sensory information is assembled by the brain into more complex cognitive representations of the world. Her research lab focus is on the cognitive map, which is formed by the hippocampus and associated structures and underlies our sense of direction and sense of place. And this map is also used to organize memories of life experiences. I was very fortunate to meet Professor Jeffery just, I think, 3 years ago. And it's fascinating what they do in their lab and the research she's carrying on at UCL, and it's a real pleasure to have her here today. So without further ado, Kate, let's start with your presentation. Thank you very much for being here today.
Thank you very much. Okay, so what I wanted to do, I mean, it's a kind of a grandiose title for my talk, but what I wanted to do was to introduce you to the cells in the brain that we've been studying that seem to be creating this cognitive map that Abba mentioned, that seems to create some type of internal representation. So when you think of a place, you're activating, as far as we know, this system. And in the last few years, we found out some amazing things about how this is organized and how the cognitive map's created. And so what I wanted to do is to talk you through some of those and then touch on why that might be relevant to architecture and to designing space. So that's something that I've become quite interested in. So it all began with the discovery of these neurons called place cells, which was a discovery made by John O'Keefe, who was working at UCL. He still works at UCL. And he was studying neurons in the brains of rats. So this is the brain of a rat. And he had electrodes implanted into this structure here called the hippocampus, which the whole structure is the dark blue region, which in the rat is quite a big chunk of the brain. And you can see a little kind of cross section, like a slice of Swiss roll, shown in turquoise here. So this is the cross section. And what he was doing is he had his electrodes that were kind of resting in this layer of cells here. And this layer of cells is made up of neurons that look like this. So, this cell is about the width of a hair, really, really tiny. And the electrodes that he had in the brain were also about the width of a hair, so equally tiny. And they can pick up the electrical activity of a single neuron. So this was quite a technological advance, being able to not just record large amounts of brain activity all at once, but actually to pick out the signals from a single neuron. And what he found was that if you note where each of the electrical impulses occurs as a function of where the rat is in space, in its environment. So if you mark each of the action potentials with a little red square, you can see that that they're all congregating in the southeast corner of this tabletop, that the surface that the rat was walking around in. And that was a typical finding for one of these cells. So different cells would have different places in the environment where they would like to fire. So if you build up a profile, this is now looking down on that environment from above, and the black line is the kind of cumulative total of all the places where the rat walked. And the red squares are the cumulative total of all of the action potentials that this one neuron fired as the rat had explored for 10 minutes or so. You can see that it's created a sort of a hotspot in the southeast corner of this environment, which I'm going to show with this kind of schematic. So we call this a place field. This is the place where the cell likes to fire. So this is a kind of a dynamic version of the same thing. So this is tracking the rat, and every time it goes, in this case, into the northwest corner of the box, the red cell fires action potentials, and whenever the rat is in the southeast, this other cell is firing action potentials down here, which I've color-coded in green. So this is to make the point, well, 2 points. One is that the cells, there's no apparent relationship and we're pretty confident of this, between where the cells are in the brain. So these cells would have been right next to each other because they were detected by the same electrode. But you can see that the places where they're active are on opposite sides of this environment. So there's no connection between the relationship of the cells to each other physically and the relationship of the place fields, the places where they fire in larger scale space. So it's a kind of a more abstract code. It's not a pixel map of place, but it's some type of abstract code of place. So O'Keefe called these place cells. And it took a while for the importance of this discovery to become apparent. People didn't really believe it at first and thought place was too much of an abstract thing for the brain to be encoding. At that stage, we thought of the brain as making a pretty kind of a flat set of associations between what comes in and what goes out. And the idea that there was somewhere in the brain that was making a map didn't really fit well. So it took quite a long time for people to really start to accept this idea. But eventually they did. And then the question became, well, how does a place cell know where the rat is as the rat's walking around? What information does it get? And the sensible thing to do seemed to be to record from parts of the brain that send a lot of projections to the hippocampus. So O'Keefe was recording here from this kind of top end of the hippocampus. And the part of the rat brain that sends a lot of connections to this part is the entorhinal cortex, which is shown here in green. And so a few decades later, in fact, this group in Norway called the Moser group started recording from this region of cortex, which is called entorhinal cortex. And they made this quite unexpected discovery, which is that the neurons in this region of the brain also fire in spatially localized regions. And they're quite reproducible. So every time the rat goes back into that place, the cell will fire there again. But instead of there just being one place or two, or, a few like place cells have, there would be lots and lots and lots of them, and they were spread over the surface of the environment in this very regular array. So this is one cell. So it's the same type of scheme that I showed you with the place cell. It's just one cell, but it fires when the rat's here. It also fires when the rat's over here, it fires when the rat's over here, when it's over here, and so on. So, and the rat's going backwards and forwards between these places. It's moseying around in a kind of a random way. So the cell must be somehow tracking the path of the rat and keeping some type of metric representation, a distance and direction representation of where the rat's walking, so that every time it goes back into one of those places, it knows it's time to fire again. So this was a really amazing discovery. Like, it was one of those moments that most of us in the field remember vividly when we first saw this very, very regular polka dot pattern produced by these cells. It was just extraordinary. And the thing that was immediately noticed is that there's a kind of a hexagonal symmetry to the pattern. So this is a pattern that's well known in mathematics called hexagonal close packed. And it's the pattern that you get if you try and fit round things as closely as possible together. They will arrange themselves into this very orderly arrangement where each of the units, whatever it is, a tennis ball or an orange or a circle or a grid cell field, whatever, is surrounded by 6 others of the same size. So you can see here in the schematic, so it forms a kind of hexagonal pattern, and it kind of tessellates the environment. So you could stack as many of these hexagons together, and they would go as far as you can get to until you reach the boundaries of the environment, basically. So that told us that the place cells are getting information about the distance and direction that the animal has walked from these cells. But also the regularity of the pattern suggested that, it's very grid-like. The fields are kind of aligned in these nice rows. And so that suggested that maybe indeed the function of these cells is to create a grid for the place cell map, so that as you're walking around, you have a kind of a scale in your brain that's telling you how far you've walked. And these grid cells come in multiple different scales. So as you record from deeper and deeper regions of the brain, you find that the distance between these fields and the size of the fields increases, so the whole pattern gets much, much bigger. So it looks like there's capacity for these cells to represent space at a variety of different scales. So this really set the seal on the idea that the brain is creating an internal representation of space. And so O'Keefe and the Moses shared the 2014 Nobel Prize for the discovery of place cells and grid cells. So it was really transformed psychology, this idea that the brain can do these abstract things. And so I've been sort of thinking, I feel it sort of has ramifications that go beyond just psychology, and in particular, those of us who think about the built environment potentially have something to learn from the discovery of this system in the brain and the properties that it has. So we've done lots of experiments of that kind of prowling around in the parts of the brain that project to the hippocampus and trying to find out what's going on. And we found lots and lots of different cells that all seem to be doing slightly different but related things to do with spatial processing. So this reference that I put in the bottom right here is my postdoc, and I wrote a kind of a summary of what was known in 2017, but new cells keep coming along all the time. But the ones that are really particularly seem very logical and salient for a map are these ones. So we've got the place cells, which I've just told you about, and the grid cells. There are also cells called head direction cells that are found in other regions of the brain, also close to hippocampus, that become really active when the rat is facing in a particular direction. So in this case, every time the rat faced to the southwest, the cell would massively increase its firing rate. But if it faced in any of the other directions, the firing rate fell to close to 0. And different cells of this type have different directions where they like to fire. So one cell might be southwest, another one might be north, another one might be east, and so on. And that pattern, if you've got a whole bunch of cells that are all kind of in the one rat, the pattern will be the same wherever the rat goes. So if one cell fires when the rat faces one direction, the relative angle that the second cell fires will always be the same. The absolute angle in geocentric coordinates might be different. So it might be north in one room and east in another and so on, but the relative directions are all the same. So these cells like to kind of maintain this coherent representation of direction. And we think that this is a compass, but it's a local compass. So every time you go into a new space, your compass system sets its orientation and then remembers that orientation by learning about the landmarks that can be seen from within that space. So it's been a really good model system for studying not just spatial processing, but also memory. So this system is also very involved with memory. And then the other type of cell that seems very logical for a spatial system are these cells that become really active when the animal goes near the edges of the environment. So we call them border cells. And the edges seem to be very important, partly because they constrain the movement of the animal. So they have, a lot of significance, behavioral significance. But also when the rat bumps into a wall, it kind of knows exactly where it is at that moment. Whereas if it's in the middle of a big open space, there might be some uncertainty. So these border cells seem to have the function of kind of anchoring the map, and they provide the reference frame against which the place and the grid cells can kind of track their movements. So the movement tracking actually is one of the really important things about this system, the way that there's this constant interplay between the movements that the animal is making and the static features in the environment. That's possibly the main take-home message of this talk, because this really, really important interaction, which is going on all the time, like every moment. The brain is going, what movement did I just make and what can I see? And do those things seem to make sense? Do they fit together? And if they do, that's fine. I know where I am. And if there's a mismatch, something's gone a bit wrong and I need to decide what. Constant kind of recalibration of position. So how do, so I said, how do the place cells know where to fire? And they seem to use the grid cells and the head direction cells, but how do they know where to fire? So there are a few kind of principles. I just mentioned one, which is that the boundaries seem to be important. And we can show this in a variety of different ways. The simplest one, and one of the first things that we did back in O'Keefe's early days, was to show that if the rat is in a bounded environment and you move that environment, so you move the tabletop over a meter or so, then the place cells will shift where they fire to follow the box. So they won't remain anchored to the room, they'll follow the local box. So the local boundaries seem to be really important. Another important relatively early finding is that if you deform the box, if it's a really familiar box, so the rat is very, has a lot of experience of that box, and the cells have learned the relationship between where they're supposed to fire and the boundaries. If you then stretch the box a little bit, the cells try to maintain the same relationship to the boundaries, but that places them in conflict with the inputs that are tracking the movements of the rat, because there's further that the rat can walk now that the box is stretched. And that seems to create a little bit of a conflict. And the cells, at least at the beginning, find a compromise. So they will stretch themselves out a little bit to follow the stretched walls, but they won't stretch themselves out completely, because they're also getting this movement information that says, hang on, you're not supposed to walk this far, shrink up a little bit. And so what we see is the outcome of that conflict. And different cells resolve that outcome in different ways. So the closer they are to a particular wall in the place where they fire, the more likely they are to be influenced by that wall. So that tells us that the environment, that the boundaries are really important, but also that different cells are processing different aspects of the boundary. So it's not a really monolithic, inflexible map. It's kind of stretchy in a way. A similar thing was found with grid cells, and this was an experiment done in my lab by Caswell-Barry, who found the same thing, that if the environment is new to the animal, then a grid cell will make a nice regular pattern. So you can see a regular pattern here, and it would be a regular pattern like this even if the environment was rectangular. If you placed the animal into it for the first ever time, it would have this regular pattern. But if you then stretch the box, then the pattern deforms. So the grid cells also seem to be learning about this relationship between the environment and the self-motion cues. Now, there is another kind of quirky finding, which we're still trying to understand, which is that it's only true that the pattern will be regular if the environment is somewhat symmetrical. If the environment is not symmetrical, for example, in this trapezoidal environment, then the pattern from the get-go is a little bit deformed, and we're not fully sure how to explain this. So it's a bit of a puzzle, and does kind of shed light on the fact that these cells are integrating multiple different types of information, including possibly expectancy. So it might be that the rats were used to living in rectangular spaces, and so when they got placed in a new space, they thought, that was the only thing they'd ever known in their whole entire lives. So maybe they thought this should be a rectangular space, and that cognitive information was also feeding into the grid cells. So there's a lot of interaction between different brain systems that's getting weighed up here. Boundaries are so strong that they can overrule path integration. So if you've got a bunch of repeating rooms and they all look the same, then even if the rat can walk from one to the other, like out this little doorway and along this corridor and then into this next room, that doesn't seem to be enough, at least at the beginning, for the place cells to incorporate that information and go, okay, I'm in a different place, so I need a different map. They seem to replicate the map. That's also true for grid cells at the beginning, but in this experiment that was done, so Caswell-Barry, who did the stretchy box experiment in my lab, then went off and set up his own lab, and the first experiment that came out of that lab was with Francis Carpenter. So they found that initially the grid cell treats these two compartments as if they're the same compartment. So the pattern is the same, even though the rat can walk, along this corridor. And even though if the grid cell was really tracking distance in a completely, if it was completely dominated by self-motion cues, and that was all it knew about, then the pattern should have been slightly different in the two boxes. And indeed, after time, that is what happened. So once the animal had become really familiar with the two compartments and the fact that the rat had to walk between them, then the pattern adjusted. So there was plasticity, there was learning in this system. So the system can learn. So the second thing is that direction is important. So I mentioned the head direction cells, and they are probably the thing that enables a place cell to fire asymmetrically, even in a symmetric compartment. So even if you're in a square room, you can kind of tell one side of it from the other. So this was an experiment that was one of the first experiments that I did with place cells when I was a postdoc in John O'Keefe's lab. And what I did was to place the rat in a container and turn it around very, very slowly so that its sense of direction was reoriented with respect to the outside world, but the rat didn't know that. And then when I put the rat back into the box, the place cells had rotated the place where they are active along with the sense of direction. So that was kind of proof, if you like, that the internal sense of direction is what is at least one of the things that orients place cells. And that's one of the big take-home messages for today that I'd like people to kind of take away, is that the sense of direction seems to be really foundational for the cognitive map, because the place cells need to know which wall is which if they're going to be able to figure out where to fire. And I think, you know, the experience is quite common where you find yourself kind of coming into a place that's familiar, but you were coming into it from an unexpected direction. And you take a long moment thinking, everything looks familiar, and yet I don't know where I am. And then something feels like it rotates in your head and it goes clunk. And then you go, oh, it's because I've come in the north side of the square and not the west side like I normally do. Oh, now I know where I am, type of thing. So I think what's probably happening there is that the place cells don't know what to do because what you can see doesn't line up with what your sense of direction is telling you. And then the brain figures it out, and then suddenly they know what to do, and you get that clunk feeling. The place cells adopt the map that they're supposed to adopt. So the third type of information that's important is what I call context, which is stuff in the environment that's not specifically spatial, but it defines the space. So for example, the color of the walls helps you know what room you're in, you know, together with all of the other things like knowing what room you were in and how you just walked and so on and so on and so on. So these non-spatial cues will also reorganize place cells. So you can place a rat into a container and record the place cells, and then you can take it out, replace that container with one that's identical but a different color or different in smell or something like that, and put the rat back, and the place cell will, what we call, remap. It'll do something different, as you can see here. So I just wanted to touch briefly on some of the investigations that we've been doing to look at how this map is affected by the structure of the environment. So I've talked about really simple environments, but the real world is a bit more complex. So it's often multidimensional. So for example, an environment might be one-dimensional, it might be two-dimensional, so that you can you know, move in X and Y, as it were. Or it might be what we call volumetric, so it fills space in X, Y, and Z. And so there's this kind of interesting question, what are the places in the grid and the head direction cells going to do in these different dimensionalities? In most environments, there are also intermediate dimensionalities. So for example, you can have an environment that's one-dimensional if you look at it locally, but three-dimensional if you look at it in the larger space. So we say that this is a one-dimensional manifold embedded in a three-dimensional space. Here's a 2D environment embedded in a 3D space. So at any one moment, the rat can only go in walk in 2 dimensions, but there are 3 dimensions available to it globally. And here's another embedded one. This is a spiral kind of staircase. So there's been a lot of work, and I should say that natural environments are usually mixtures of all of these things, and it also depends on the sort of ecological sort of ethology, if you like, of the animal that you're looking at. So some animals can fly and swim, others can only crawl, and so on. So we've done some speculations about what you might expect place and grid cells to do in these different environments, so that we know what the pattern is on the flat. The place cells kind of have these flat place fields, and the grid cells have these flat grids, if you like. But what happens if you're in a volumetric space? Will you see a completely volumetric pattern where the fields are actually globular, and it's as if the flat ones are a cross-section through a pattern that in 3 dimensions would be globular, if you like. Or it might be that the system doesn't really track vertical travel. It's really only interested in how you're moving relative to the surface of the Earth. And so it might be that the pattern for place cells would be the same at every level. In other words, in 3 dimensions, it's kind of, you have these kind of columns, and same with the grid cells. Or it might be that you have a sort of a pattern in 3 dimensions, but it's a bit kind of stretched, so there's a lower resolution. So with place cells, so I'll talk you through these pretty quickly, but just to make a couple of points. In 1 dimension, place cells do something different going one way than they do coming back, which is slightly surprising because that's not what they do in 2 dimensions. In 2 dimensions, it doesn't matter what direction the animal is traveling through a place field. And we think that this is because in one dimension, the hippocampus has decided to treat this as if it's two different environments. In this 1D environment embedded in 3D space, we thought that we might see a different pattern in the sloped version of this track than in the flat version. But actually, what has been reported is that the place fields sort of either follow the track when it's elevated into the third dimension, or they remap, they do something entirely different. So some do one, some do the other. And it sort of seems like the rat is trying to encode both things at once. It's encoding both the similarity in the two situations, but also the difference. And indeed, we think that's one big function of this system, is to work out, if you go into a room and it looks a bit different, is that because it's not the room you thought it was, or is it that someone's redecorated or shifted furniture? So the hippocampus is always trying to decide, is this the same thing that I already know, which is called pattern completion, or is it something different, which is called pattern separation? In this 1D embedded in 3D, we saw with place cells that they fire kind of close to each other in 3D space, as if the brain has worked out that those locations are close to each other in 3 dimensions, even though they're quite separated in 2 dimensions. And if the rat is climbing around on a vertical wall where it can stand on pegs, so it's oriented horizontally, standing on these little jutting pegs, but moving around vertically, then we get relatively normal looking place fields. So the system can track, it is capable of tracking movement in the vertical dimension, which was kind of interesting. If the rat instead walks around on a vertical surface by clinging to the wall instead of standing on pegs, so now it's oriented perpendicular, again, we do get place fields, and they look normal, but there are fewer of them. So, which is slightly surprising, and we haven't quite figured out why that is yet, but we think it's because the system is struggling a little bit with this unusual dimensionality, and so not as many cells are getting excited enough to fire as you would expect. And then place cells in a volumetric space where a rat can walk through a lattice, they produce 3-dimensional globular place fields. So that's proof that the system can track location in 3 dimensions. So the grid cells is something slightly different, but there are also some interesting, slightly similar, but there are some interesting differences. So as with the place cells, we get a different pattern going one way than coming back the other way. But we do see a pattern that's consistent with being a slice through a 2-dimensional pattern. So it does, if you do the statistics on the places where the cell fires, it is consistent with being a section through a hexagonal pattern. In 2 dimensions, I've shown you that there's a hexagonal pattern. On a sloping surface, we thought we might see that the pattern becomes different because we are cutting through a 3-dimensional pattern at a weird angle. But actually, it looked like what we saw with the place cells, which is that the cells just seem to have followed the surface as if they don't even know that it's tilted into the 3rd dimension. On the spiral staircase, unlike with the place cells, so the place cells seemed to be tracking distance vertically because they were able to fire in a localized region of vertical space, but the grid cells don't seem to do that. So they fire on every coil as if they don't know that one coil is different from another. So it seems to be that they are not getting information as the rat's going around and it's being elevated in vertical space. The cells are not tracking that. And we wondered whether that meant that they weren't tracking vertical distance at all, because we saw something slightly similar on the pegboard. But when we did the experiment on what we call the chicken wire maze or the floor wall maze, where now the animal is able to kind of walk around on the surface, we did see something a little like a grid pattern, although it was a bit distorted. It was bigger and it wasn't as regular, but certainly there is now some capability of tracking distance in the vertical dimension. So we think that from the point of view of the grid cells, it matters how the rat is moving through the space, whether the rat is walking in the plane of movement or whether the rat is moving orthogonal to the plane that its feet are on, which was quite an important sort of realization for me anyway. And then in a fully volumetric space, as with place cells, we do get the firing extending into all of those dimensions, but it's not regular anymore. So it seems to be irregular. So we think that the map has a different metric foundation if you're able to move in 3 dimensions. And indeed, in all of these different types of apparatus, the map seems to be slightly different. So the conclusion seems to be that the map that's being built up is very much arising from an interaction between what's out there in the world and the movements that you're able to make through it. So just to quickly finish with a couple of implications for architecture. So one thing I've wondered about, and this is not a serious proposal, it's merely a hypothesis, because I've mentioned this once to someone else and they said, oh, these are the reasons that it can't possibly be that. So it's just an interesting thing to think about. Is there something special about hexagonal symmetry in the environment, given that the metric foundation of the cognitive map seems to be hexagonal? And there's been a kind of remarkable lack of hexagonal symmetry in architecture. And I wondered whether we have an aversion to it. And I found this paper that has kind of gone through the literature on hexagonal design over the past century or so. And there have been some attempts to design hexagonal city layouts and things, and they've never really come to anything. So it's interesting to speculate that maybe we dislike hexagons because they are too kind of similar to the underlying structure of our map and therefore not interesting or something like that. Or maybe we just haven't, maybe it just conflicts with our requirements for engineering. So I wouldn't want to say I have any opinions on this, but I think it's interesting to think about. Second thing is the role of the sense of direction in design. And I think that this is something that's come about from studying the head direction cells. And we've got many studies of head direction cells and their properties now. And at least in rats, we've got a really good idea of what the cells can and can't process. And it seems to me that architecture could learn from that. And one of the things it could learn is that rotational symmetry is difficult for the head direction system to resolve. And as I mentioned, if that then conflicts with your place cell map, it might make it hard to organize your cognitive map. So I think you need to be very careful about circular symmetry. And then the third thing is, how do the movement cues in a space affect perception of the space? Because the one thing that's really certain about the structure of the cognitive map is that there's this constant integration of the movements that you're making and all of the signals that generates, including vestibular signals and optic flow and all of that kind of stuff. How does that interact with what you can see and what your senses are telling you about the space in the outside world? And I think there's a lot of potential in architecture to play with this relationship and to exploit it, to confuse it, to assist it, like all of these things. So I think thinking about movement in relation to the self-motion system, which is pretty well understood now in the brain, is interesting. So I'm going to just stop there. I'll put up these take-home messages and I'll just leave them there for you to read. And I guess we haven't got much time for questions, but we have a panel discussion at the end, and I'm really happy to discuss if there are any things that people want to raise there. So thank you for your attention, and I'll hand back to Agon.
Thank you very much, Kate. Fantastic. I have a list of questions here. It's one enigma after the next. It's really, really interesting.